Good morning, everyone. A warm welcome to all of you for the Somaiya public lecture. To begin the session, may I request our Pro Vice Chancellor, Dr. Satish More, Dean Academics and Research, Dr. Santosh Narayan Khedkar, Dean Science, Dr. Nitin Desai, and our speaker, Dr. Chetana Sachidanandan, to kindly proceed with lighting of the lamp. I now kindly request Dr. Satish More, Dr. Nitin Desai, and Dr. Chetana Sachidanandan to come on the podium. I request all of you to rise for the campus prayer.
ईशावास्यमिदं सर्वं यत्किञ्चित् जगत् तेन त्यक्तेन भुञ्जीता मा कृतस्य कृद्धनम् मूकं करोति वाचालं पङ्गुं लङ्घयति गिरिम् यत्कृपातमहं वन्दे परमानन्दमाधवम् ॐ प्रणोदे सरस्वती वाजेभिर्वाजिनीवती वीणामविद्यवतु ॐ
May I request the Pro Vice Chancellor, Dr. Moore, to say a few words?
Very good morning to all of you and welcome to this public lecture. Today we have with us Dr. Chetana Sachidanandan, who is a senior scientist at the Institute of Genomics and Integrative Biology. It is a CSIR-Institute in Delhi. About Somaiya Vidya Vihar University, it is one of the first private university in Maharashtra. It is a self-financed university, and in 2019, various institutes within the Vidya Vihar campus like Management College, Engineering College and others, they came together to form a university under Maharashtra. It is recognized by UGC and recently it has received NAC A accreditation. The university offers various programs. There are 13 faculties. Other than engineering and management, we have a School of Commerce and Business Studies, School of Basic Sciences, School of Humanities and Social Sciences, Maya School of Music, and the best feature of the university is the Faculty of Dharma Aesthetics, which is very unique for the university. So the idea is to provide a holistic education to all the students who are taking admission in the university. The sports academy is one of the very big salient feature of this university, where we have swimming pool which is of the Olympic size, and the squash courts is also very well recognized, and our students have won international competitions, and I think recently we will be organizing all India level squash championship also within our campus. So this is a brief introduction about the university. All of you know about it, but still it is better to remind us where we stand. This public lecture is one of the series where we invite eminent faculties, professors, scientists, industrialists to share their views and experiences. This is a great opportunity for all of us to listen to such public lecture and increase our knowledge. So this is a tradition we are part of. Again, welcome you all for this public lecture. Thank you.
Thank you, sir. I now request Dr. Nitin Desai, our Dean of Science, to introduce and welcome the Somaiya Public Lecture speaker, Dr. Chetana Sachidanandan.
Good morning, everybody. It gives me immense pleasure to introduce today's speaker, Dr. Chetana Sachidanandan. She is a senior scientist at CSIR-Institute, already or Pro Vice Chancellor has said, at Institute of Genomics and Integrative Biology, IGIB, South Campus, New Delhi. She is also professor at Academy of Scientific and Innovative Research at Delhi. She is also cadre at AcSIR. After completing her MSc from JNU, she did her PhD from CCMB, Center for Cellular and Molecular Biology, Hyderabad, and then she shifted for a postdoc at Cancer Research Institute, UK, London, and then at Massachusetts General Hospital and Harvard Medical School, Boston, USA. After completion of postdoc, she shifted back to India as a Ramlinga Swami Fellow by DBT. Until today, she is with IGIB as a scientist, senior scientist. She is member of various societies, to name Indian Society for Developmental Biology and British Society for Developmental Biology, UK. She is on editorial board of various journals, specifically Scientific Reports by Nature Publishing House, London. Apart from that, she also writes very important as a scientist to create awareness among the community too. General articles in newspapers, especially Hindustan Time, Indian Express, on topics like anxiety and child world, helping dyslexic children and bloom, opening arms and mind to autism and diversity. These are the very important sensitive issues when scientists speaks that penetrates deeply in the society. So such a great job she is doing. And scientific contribution wise, she has published more than 30 research papers in different national and international journals. So we have such a outstanding researcher, scientist, and a social blogger where she does blogging for science and readings. So such a all-rounder personality is with us. So I welcome her, and I look forward for her talk on Zebrafish as a model for human diseases. Thank you, madam.
I now request Dr. Chetana Sachidanandan to deliver the talk. Hello everyone. Hi. So I know that it's on the YouTube and all, but you are all sitting here. So it would be great if you interrupt and ask me questions, because I know that this is a diverse audience and I might get too technical. It's a tendency that we have. So do stop and ask questions if you have, or you can hold it later and also do that later. I will try to introduce this system that I'm in love with, and I hope some of you will also fall in love with it. I have met some people already in the university who have experience working with zebrafish, and believe me, once you look through the microscope and these embryos, there's no way you can resist working with them. So zebrafish, and my sort of emphasis today is going to be how you can use this model as a model for human diseases and to study embryonic development through different strategies. So before we launch into that, I want to introduce this beautiful fish to you, and it is used globally for research on embryonic development, on diseases, as a toxicology model and many other systems, but it actually originated from here. And this it is working here. Oh, okay, take it. So that's fine. I mean, there's not much detail there. It's basically a map of India showing where all these fish have been collected from. So this is from a paper where basically Dr. Pariche has collected these fishes from various parts of India, and they look slightly different from each other, as these pictures show you. But they are all Danurario and other species of the same genus, and very related. They live in these tropical climates like ours. And the reason we biologists love this fish is multifold. One is that these are very small embryos when the egg is laid. So unlike the mammals, where the fertilization happens in utero, right, inside the mother, in fish, frogs, etc., the fertilization happens outside. So the eggs are laid by the female, and the sperm is released by the male in the water, and the fertilization happens there, which means that if you control the condition, you can have access to the embryos from the first cell stage onwards, from the zygote cell stage onwards, which allows you to be able to watch development in real time with these embryos. And as you see in these images here, these embryos are transparent, and they are big enough for us to put under a regular stereozoom or a dissection microscope that we have in our college laboratories. So you can watch these embryos over a period of time and see the developmental process. The male and female, as it is shown in this picture here, are slightly different. There are some organisms like the peacock and peahen, for example, very dramatic differences between male and female, right? Here the difference is such that it is very easy to get confused. So when students start setting up matings of these fishes early on, they make mistakes because the male and female look very similar. The female is slightly lighter in color and has a bigger belly because this is where the eggs are stored before they are released. The organ systems inside the fish. So I'm showing on the top is a larva, and the bottom picture is an adult.
and they have all the organs that we have except one, which is basically the lung, right? These are aquatic organisms, but every other organ that we have is present in the, a good complement of it that is present in the fish and does similar kind of functions, and very importantly originates from similar kind of cell populations, and similar kind of genes control their development and function. So looking at any of these organ systems in the fish is can tell us stories about what happens inside us humans, right? These are the people who made the fish a great model organism, because when you start, so the first picture there on the left hand side, the black and white picture, is George Streisinger, who went to a pet shop and basically asked for some fishes that are robust and can be bred in the lab, and you can kind of maintain them. And the pet shop guy picked up zebrafish and gave it to him. If you go to a pet shop here in any place in India also, you will find zebrafish in the aquarium there, because these are very sturdy fishes. They don't need much sort of gentle taking care. And if you manage the temperature and light and all that carefully, you can also have them breed in captivity easily. That was one of the criteria that Streisinger had, because he wanted to look at the development of the embryo. So many of you might be familiar with Drosophila, the fruit fly that we see at home all the time, but which is a staple model organism for developmental biologists. And Drosophila opened up, it's a fly, so different from us, invertebrate. I mean, what has it in common with us? But Drosophila studies have illuminated so much of embryonic development for not just human beings, for all sorts of organisms, because the basic principles remain same, right? So Drosophila was the organism that opened up embryonic development at molecular scale for us to study, and from there zebrafish kind of took on this job and made vertebrate development easy for us to understand, easy for us to access. And the three people in this image on the right, top right, is Christian Nusslein-Volhard, Nobel Prize winner, and Mark Fishman and Wolfgang Drewer. These three people basically set up a genetic screen in their laboratory where they created large number of mutants in different genes in the fish that caused problems in embryonic development, right? I will talk about that. Basically what they did is called a forward genetic screen. Forward genetic screen, the idea is that I don't know anything about the genes that are important for making heart or kidney or brain or eye or whatever, but I can see these organs, I can observe them, right? So I start with an interesting phenotype. So if you remember, Drosophila has this red eye, white eye phenotype, right? Similarly, you can pick up the organism that you are studying, whether it is mouse, which I've shown here, or zebrafish, and you look for interesting phenotypes, or basically structural changes or something that is very obviously different in the animal, right? So the way you do these screens is you, I don't know if I have a, yeah. So you take animals and treat them with a carcinogenic drug that makes changes or makes mutations in the DNA. These are dangerous molecules, of course, right? You are familiar with what carcinogenic means, cancer causing. Why do they cause cancer? Because they create mutations in the gene. So, but we can actually use this for studying biology also. So you treat animals with these molecules, and these molecules go and randomly do changes in the genome and mess up with genes that are in the genome, right? But some of these changes in the genome may have no effect on the animal. You may not be able to see anything, but some may have dramatic effects. So you then breed those mutant gametes. So when I do the treatment in the first fish, only the gametes in that fish are useful to us, because that is what is going to take this change forward into the next generation, right? So when you cross these fishes with other animals and you take the next generation, you now have animals with mutations in certain genes. You breed them out again, outcross them, and then incross them to bring it together to get a homozygous condition, which is not very, if it's technically complicated to understand, it's not important for us to, for the story that I'm telling. The basic idea is that I changed genes, I got fishes which have some kind of problems in them, right? So we go back to this. So there are some changes. It could be that brown fur color has become white, or could be that there is no eye, or there is only one eye, or the heart is not functioning, or the pigmentation stripes on the zebrafish is looking different, right? Then you go from there to the gene. So with the complicated linkage analysis that people used to do in older times when we did not have access to molecular biology tools, we would keep crossing the animals to figure out where this change in the genome is and identify that gene which has a change. Now what you have done is you have connected the phenotype with a gene. This is the holy grail for molecular biology for all of us, right? All diseases, all great abilities like musical ability, artistic ability, tall, short, hair color, everything that we study as related, as affected by genetics, we are always trying to figure out which gene is responsible for this characteristic, right? So that is the kind of connection that you draw when you do a forward genetic screen. So in this process, I discover a gene that is important for the fur color of the mouse. That's what the ultimate goal of this kind of a study is. Now, so I was saying that these three did this kind of a screen and found, this is just an example of one of the characteristic, which is the pigmentation on the fin. And you see the variety of pigmentation that you see on the fin of the zebrafish, right? Caused by mutations. And now you can go back and figure out these genes and what they are doing to the pigmentation. So they might be important for pigmentation, they might be important for the distribution of cells on the fin, they might be important for the death of certain cells that are making pigment, right? So all these can have the result that you're seeing on the screen here. So this was how the forward genetic screen kind of exploded zebrafish into the field of developmental genetics, and how biologists pick them up to study further. As this was happening, there is a huge revolution that happened in biology, right? I don't have to introduce the idea to you. You are the younger generation which was born into the world which already had the human genome sequenced, right? So now we have the human genome and many other organisms' genome sequences available to us on the click of a button, right? So here I am showing you in the paper where in 2013 the zebrafish genome sequence and annotation was published. This was from the Wellcome Center in Sanger Center in UK. At the same time, the sequence of the Assam, the zebrafish caught from the wild in Assam also was sequenced at my institute, CSIR-IGIB, by Dr. Sridhar Shivasubbu and Vinod Skaria. So now we have this wild sequence also available and the laboratory strains also available, and now we have a full complement of the genome sequence. I am not saying the full complement of genes, because that's still every day we are discovering new genes that are coded in this genome, and our limitation is our understanding of how the genome works and what we call a gene, right? So that's another story to tell for another time. So with this revolution that happened, that when the genome sequence became available, I did not now have to start with figuring out the fur color and walk my way back to the gene, right? I could start with the gene. And now with the genome sequence available, I also have knowledge of multiple genes that are implicated in diseases, right? So we have now patients whose genomes have been sequenced and mutations identified in them. People, so large scale studies are being done to figure out how our skin color is genes that are to do with our skin color, for example. So huge genome sequencing studies to identify what causes diabetes. So we have a surfeit of genes with suspected functions in our hand, right? So now I can start my study the other way around. I can go in the reverse, yeah, from the gene to the phenotype, right? So now what we do, so most of you who might enter into biology at this stage would not be going from in the forward genetics fashion, but rather you will be thinking in a gene-centric fashion. That I have this is my gene that I want to study, what does it do? So I would start with perturbing that gene, mutating that gene, right? So this is an example of how you can change the activity or the function or the expression of that gene to study what that gene does in an organism. So what I'm showing you here is something that we call knocking down genes, which is different from mutating. So what I mean by knocking down genes is I reduce the level of the active gene product that is there in the animal, okay? And how do I go about doing it? So in zebrafish, this is something that we do very regularly. This is the bread and butter of trying to figure out what gene functions are. So we use antisense oligonucleotides. So the messenger RNA that is there, the mRNA, is coding for a protein, right? When I take a small nucleotide, an oligonucleotide that is complementary to that messenger RNA, that oligonucleotide will now go and bind to the mRNA, the ATGC pairing that happens in DNA and RNA, right? This fact now can be utilized to block the function or expression of that gene. So the way we can do that is, for example, what I'm showing you in the left hand side of this picture with the line diagram is that I have designed an oligonucleotide which is complementary to the site at which the protein translation starts, where the ribosome binds, right? So now this oligonucleotide is binding to this region of the mRNA, and because it is binding there, that sequence is no longer available for the ribosome to come and recognize and bind and start translation. So the translation of this mRNA gets inhibited or blocked. Similarly, I can block the splicing of the mRNA. So you must know that mRNAs are cut and then pasted together to create functional mRNA from which the protein is coded, right? So there are sequences called introns that are removed from there. So this process can be blocked also using oligonucleotides. So we can in this way interfere with this protein's expression and then look at what, if I do that to the gene that I'm interested in, what happens to the fish, okay? So I'm not going to go over this figure. This is just an illustration, but I will talk about it in a few slides down the line. You can also, of course, make mutations. And in today's world, we don't make mutations with carcinogenic compounds. We do very targeted mutagenesis, right? By something called CRISPR-Cas9. I see some nods in the audience, so I guess you must have heard about CRISPR-Cas9. This is a genome editing tool. Similarly, there are other genome editing tools. So what genome editing tools do is, it gives us control over which gene and where in the genome I want to go and make a cut in the DNA. When I make that cut in the DNA, now the DNA will try to repair itself in the cell, right? It has to, otherwise the cell will die. While repairing, it makes mistakes. Those mistakes then become mutations that we can use to study the gene's function, right? So these are genome editing tools that I'm showing you here, which result in mutations in the genes of our interest, and we can then go on to figure out what that gene, so it is equivalent to, so genetics is equivalent to, if I want to understand how a car functions, I can take out parts of the car one by one, right? Take out the steering or take out the wheel and see what happens to the car's basic activity. And in that process figure out what the steering was doing or what the wheel was doing or what this button is doing. Similarly, you take out the gene and then see what happens to the organism. That tells you that that gene was important for this process that I am looking at, right? So this is how genetic studies work. So combining genetics with other kind of tools that zebrafish allows can be very powerful. So another such tool is live imaging. So fish, like I said, the embryos are transparent. That allows us to actually put these embryos under the microscope, and live embryos in water under the microscope, and observe them over a period of time. And these are the larval fish on the top, which is transparent, and that's the adult fish with pigmentation pattern, which gives the fish its name, right? The zebrafish comes from the black and white. It's actually not black and white, it's blue and sort of yellowish stripes that the fish have. So that's a non-transparent adult fish. But when scientists get something ready-made, such as a transparent embryo, they get greedy. They want transparent adults too. So now we have figured out how to make transparent zebrafish. This is by mutating the genes that make the pigment in the fish, right? If you mutate all the genes that make pigment in the fish, you will have a fish without pigment, where you can see all the parts of the fish now. You can actually, if you look there, those are the eggs loaded in this female fish's belly, right? And the heart is just above that, the red blob that you see. There is liver there. You can see the muscles. So the whole adult fish is transparent and it's swimming around in the water tank. And if you put it under the microscope, you can image the sort of things that are happening inside the fish. This fish was actually made to study cancer metastasis. So what scientists wanted to do was inject fluorescently labeled cancer cells and then watch how the tumor spreads across the animal. And for that, you needed a fish that does not have pigment, right? So that you can follow the movement of the cancer cells. You can also do live imaging like this in real time. So this is basically a compiled video image of the first 18 hours of zebrafish embryo developed. Okay, so that's the 2-cell stage that you start seeing when the fish development is proceeding. So the first cell division happens 20 minutes or so from fertilization, and after that there is rapid cell division happening in the early stages. This kind of rapid cell division happens in all embryos, and never in your life again will you have this fast cell division happening. So the cell division speed slows down, but at the first few hours of development we all have extremely rapid cell division happening, and that you see happening, and the cells are becoming smaller and smaller and smaller and smaller, and then they pack up, and then there is an explosion that you see of like movement, right? Until then the cells are just sort of wiggling up there, division happening, then packing up, and then suddenly they'll start moving. And now think about it from a cell's point of view. The movement is huge distances, right? It's like going from here to the United States in a boat. So the cell knows exactly where to go. Each cell knows exactly where to go, otherwise we will not be sitting here. Each of us had this kind of migration happening inside us in the early hours that led to the brain being here and the arm being here, right? If the movement did not happen properly, you would have a scrambled egg. You will not have anything that will make sense, right? And it happens in every individual, every animal that is born. So, and how does that happen? That is the magic of development. It is basically every cell has some information stored inside it, but that information is also being exchanged between neighboring cells, and they are talking to each other. So if one cell is moving this way and the other cell is moving this way, they talk to each other while they meet. They give information to each other that I'm going towards the brain, you go towards the heart, right? And that's like a synchronous swimming exercise. right? Very coordinated events. You can watch that. You can watch a live animal under the microscope with a beating heart. So this picture is a transgenic zebrafish larva with a fluorescent protein made only in the heart muscle cells. That is why your heart is red fluorescent and everything else is. So I've increased the brightness of the image so that you can see the rest of the fish. Otherwise, you will only see this beating heart under in the dark field of vision. Here, you are even able to see the atrium, the ventricle, the valves between the two in such detail that you can study heart function in a live embryo, and you can add drugs to it, you can make mutations to it, and you can see what happens to the heart function, right? So, and now with the aid of live imaging, transgenic animals and computation, scientists have now figured out amazing things, like this animal has every cell in that embryo being tracked over a time of 12 hours and then computationally assembled and plotted so that I can now make a virtual video of the cells dividing, moving, settling in different places, right? Each cell is tracked by the computer and is color coded so that I know when we started, what happened to that fifth cell on the right hand side, where did it go and what happened to it. That's the kind of resolution we have to study development. Okay, so I want to specifically talk a little bit about neurodevelopmental models, because we can imagine that the fish has heart, kidney, blood and liver and everything that we have. But is the fish brain anything like us? I mean, it's a fish, right? I mean, how intelligent can it be, and what is the use of studying fish to understand our brain? Fish is extremely complicated animal, and it has a complex social behavior structure, right? You don't need to be told, you know that there are many fishes that move together in schools. They evade predators. If you watch National Geographic, you see how when the whale tries to catch the fish, they run in formations and they escape and all that, right? So fish interact with each other, and and they have complicated brain activity. So the first image that I want to show you before we go to behavior models, this is the image of a fish thinking. Okay. So what I mean by it is that a fish larva has been immobilized, the brain is under the microscope, and I have a way, I'm not describing in detail, I have a way, this is not my work, I mean scientists have a way of detecting calcium bursts inside the cell. So when a neuron becomes active, it basically releases calcium from inside, and there is like a lighting up of calcium there, right? So every time you see this happening, the lighting up, it's basically those neurons are firing. So this fish has been immobilized in agarose under the microscope, and it is being shown an image in front of its eyes. So the fish cannot move, but the fish's tail is flipping and it thinks it's moving, and it is responding to the visual cues that it is getting, and that's what you can see in live here. This is the kind of power of live imaging that you can use zebrafish for, right? Now when we step back a little bit to look at behavior, like I said, fish has complex behavior, and these are all the kind of behavior that people have been studying in zebrafish. And you can create models of aggression, of anxiety, of social interaction, of cognition in zebrafish using different kinds of behavioral assays. So you can study neurodevelopmental disorders, neuropsychiatric disorders, genes that are important for different kinds of behavioral aspects of our sort of day-to-day life, looking at in fish, right? And with great sort of resolution, and we can get a lot of information out of it. So that's about fish as a model for behavior. So what can you do using chemical biology now. So why am I even talking about it? So fish live in water, right? You must have read news, heard the news, how water pollution is such an important problem, right? How all human habitation everywhere in all big cities is destroying the water piece. So basically, the water, the contaminants that we throw into the water is what the fish and other aquatic animals are taking up, are getting exposed to, right? And there are severe effects on them. The same concept can be utilized in the lab also to look at molecules, chemical molecules, and their effects on whether it is embryonic development, whether it is behavior, whether it is organ development, various different processes, right? So I will just give a kind of so many of the faculty here seemed interested in it, so I want to just take a few minutes to show different kinds of things that can be done using chemical biology in fish. So to draw a comparison to the genetics that we were talking about, in genetics we were trying to understand, connect a phenotype to a genotype, so the characteristic of the animal to the changes in the gene is what we were trying to figure out. In that you had to either make mutations or changes in the genome or the gene products, right? You can do that, you can do this kind of connections and studies also using chemical compounds. You could treat the animal with chemical compounds, look at what happens to the animal, then figure out which gene products or proteins were responsible for that process that we are seeing, right? Let's go to a couple of examples. So how would you do a chemical genetic screen in zebrafish? Super simple. You don't need any kind of genetic manipulation, any kind of fancy equipment. Set up crosses, collect embryos, you put them in water because fish lives in water and you can dissolve compounds in water, right? So that is why fish becomes an ideal model if you want to do any kind of chemical biology, whether it is toxicology or chemical genetics. You can expose fish embryos to different compounds in very simple way, and then you can put them into separate wells and treat them with hundreds of compounds. The number of embryos is your only limitation, right? Hundreds of compounds can be used. Then you look at what do these compounds do to the fish. So, and why would you look at that? What can you learn from there? So there are two ways in which you can use this property of fish to respond to chemical compounds. You can think about a normal fish, that's on the left, and a fish with some defect, that's on the right. So you can look at the chemicals that cause a normal embryo to become an abnormal embryo or a defective embryo, right? So those are the compounds that will cause bioactivity, like can be a toxic molecule or it can be a molecule that tells you something about development. I will give you an example in the next slide. Or you can think about it the other way around. I have a fish with a defect in it, a disease model, right? And I use chemical compounds to return it back to normalcy, which means the compound is like a drug. When we have diseases, we take drugs to get better, to become normal, right? So that's another way of looking at what chemicals can do. So let's just look at an example of figuring out how one molecule or one or two molecules can cause toxicity in fish, okay? So this is a busy slide. I don't want to confuse you. All you have to look at is the water on the leftmost corner. That's what a normal fish sitting in water looks like. The next two rows that you're seeing are two compounds that are used in battery industry and other such industrial applications, and what they do to the fish. So if you go from left to right, these are longer exposures and you see that balloon-like fish. It's basically, its heart has stopped functioning completely, and because the heart is not pumping, there is edema. So even in human beings, when the heart doesn't function properly or the lungs doesn't function properly, you get edema, right? You fill up with fluid because it's not circulating properly. That's what happens in fish too. It's ballooned up because it is now filling up with water, right? So there is a heart defect. From by looking at that fish, you can see that there's a heart defect and many other defects. The red image that you see at the bottom is a transgenic animal where the liver is expressing or making a red protein, a red fluorescent protein. So I can see only the liver. I don't have to get distracted with the rest of the animal. Only the liver is visible, and I can show you that in C and D images, when these compounds, these toxic compounds were put on the fish, the liver had massive cell death and shrunk in size, right? The green image is again a transgenic zebrafish which has a green heart, because I have made a green fluorescent protein that is made only in the heart. And I can see that from the B image, which is the normal fish, to go to the last J image, you can see that there's dramatic difference, right? You don't have to understand anything about heart development to know that there is a massive problem there, right? So these molecules are very dangerous for the liver, for the heart, for and for the functioning of the animal also. But molecules can be good also, right? They can be useful. So this screen we did to find chemical molecules. So this was a collaboration we did with a chemist to find molecules that can stop blood vessel formation. Now you must be wondering, why would I stop blood vessel formation? Blood vessels are very important, right? Angiogenesis is extremely important. But cancer, tumor cells trick the rest of the body around it to make blood vessels towards it so that the tumor can get nice blood supply, oxygen and nutrients, right? So one way to kill a tumor is to prevent the blood vessels from forming. So we are using zebrafish here as a model to get identify small molecules or chemical molecules that can prevent blood vessel formation. So the green that I am showing in the B, C, D, E and F are labeling blood vessels, and the B is the normal, how it looks, and the next the D, E, F are compounds that were derived from this synthetic pathway that prevented blood vessel formation. So you can actually identify molecules with cool effects like this, which are, this one is for example preventing angiogenesis. You can also identify molecules with powerful activities or functions on a particular pathway. So I am showing you an example, doesn't matter what BMP is, just look at these three embryos here. The first one is the normal, the two are very weird, right? The middle one does not have a head, the bottom one does not have a tail. So this is basically a pathway. So these are mutants in the pathway that makes our head tail orientation. So embryo is a round ball in the beginning. Then there is asymmetry in the embryo, and the embryo becomes like head one side and tail one side, right? Left another side and right another side. So this is what gives us the structure that we have. So this molecule called BMP is very important for deciding which is the head and which is the tail, right? So this is what the mutations in these genes look like. Now if I take a bunch of fish embryos, take a lot of compounds. So this study had 7,000 compounds that were tested. We found one compound that looked like the same tailless fish that I saw in the previous slide, right? Which means I have found a chemical molecule that is interfering with the protein and the pathway that is important for the head-tail formation. Why do I need this molecule? What do I want to make head-tailless fish and all that, right? But in biology, we have only a few small number of pathways, molecular pathways in our body. Those pathways are used again and again and again for different purposes throughout your life. So the same molecule that makes the head and tail in an embryo also makes bones in our body, also causes cancer when you're older. So what we found using this study is a drug-like molecule that is able to prevent this protein from doing its function, which can be very useful for bone diseases and cancers, right? So using zebrafish, you can find molecules that have powerful effects and you can take it to the clinic later. So this molecule is in clinical trials right now, derivative of this molecule. This just show that this molecule works like this. So just to sum up, what can you do with chemical screens in zebrafish? You can go from a normal to an abnormal. In this process, you can identify interesting bioactivities or figure out toxicity of the molecules. You can also go from abnormal to normal. I have not shown you examples of that. So from a disease to a normal condition, right? So if it's 12:20, what's okay? So then I will use one example of a research project that we have been working on for the last like 10 years or so, where I will illustrate all these things that I talked about, the great things about zebrafish, how we can use it for asking research questions, right? So what do we do at IGIB in my lab? We model human diseases in zebrafish. So that's the fish model called Oscar if you watched Shark Tank. So these are the kind of diseases that we model. You don't have to look at the pictures. Just telling you that we look at rare congenital disorders, basically babies born with birth defects, that kind of diseases. We look at liver damage and regeneration, non-alcoholic fatty liver disease or other metabolic disorders, right? So I'm going to talk about one such disease as a illustration of what you can use zebrafish for. So I'm talking about this disease called Rubinstein-Tayeby syndrome. It's a rare congenital disorder, very rare. 1 in 100,000 or so is the prevalence from Western data. In India, we do not have good registries of birth defects. We know, you all must know of people in your acquaintance around you, or have heard of people who have birth defects, or babies who were born soon after being, who died soon after being born, right? These are all because embryonic development does not sort of proceed properly. What I was saying, this amazing dance of cells that happens, if something goes wrong, you can have disasters, right? And so those are birth defects, and for us to know what is the prevalence of a particular kind of disease in our country or in our state, we have to have a record of all the births that happen and what happens to those babies, right? We, as you must know, we don't have that kind of records. There is now there are quite a few birth registries. Pune is one place where there has been real advance in making such registries for congenital disorders, and some others that have are coming up. Anyway, going back to Rubinstein-Tayeby syndrome. So it's a very rare disease. So usually, so when I say rare disease, we should also think about why are these rare. So Mendelian disorders where a single gene is mutated and causes a disease is a rare phenomenon. You will either have, you will not have birth because the defects is so huge, or you will have no effect. You might have mild mutations and you may not even know that many of us are carrying mutations in our genes that are not manifesting in any way that is visible, right? So to have one gene that can have a dramatic effect, it's a very rare condition. Any other kind of disease that you see that is very common, say diabetes or blood pressure, thyroid, these are all things that you must have heard in your family. These are not rare and they are very common because there are multiple genes involved in creating that disease, right? So they are also more complex. These are easier to study because there is one gene, one disease. So this disease is actually caused by mutations in two very related, closely related genes called CREB-BP and EP300. How did we discover that? By sequencing the patient's DNA. Multiple such cases were analyzed, and some 60% of patients with Rubinstein-Tayeby syndrome seem to have mutations in CREB-BP. some 10 or so 10% patients have mutations in the gene called EP300. That's only 70%, right? This is a disease that we seem to understand very well what gene is involved. Again, 30% is unaccounted for. So for most diseases, it is like that. We may know the majority of the cases, but there is still this mysterious 30% that we don't know what's going on. Anyway, so it's a syndrome, which means multiple systems are affected. That's listed there. The syndrome is also called broad thumb and hallucis syndrome because the thumb and the big toe on or the feet are broad or deformed, and that's one of the characteristic features. Then there are facial features that are that the doctors can classify to identify that this is the particular syndrome that we are talking about. They also have heart defects, gut abnormalities, intellectual disability, and behavioral issues. So it's a disease with a lot of problems, and also fatality in the early stages because the babies are born with severe defects. Sometimes they don't survive. The genes that are mutated in this disease, CREB-BP and or CBP and EP300, are proteins or enzymes that basically transfer acetyl group, the acetyl group from acetyl-CoA to histones. Okay, this is a process called these enzymes are called histone acetyl transferases. They are very important for controlling how our DNA is opened up inside the nucleus. So whenever our DNA is tightly packed in the nucleus, and when we need to use one part of the genome for getting information out, we have to open up that part, and that opening up is a very controlled process, and that control is done by enzymes such as histone acetyl transferases. So first thing that we wanted to do when we started this project, of course, is, so there is a disease, human disease, there is some genes known. Do these genes exist in zebrafish? That's the first question. So we went looking to see whether zebrafish genome has these genes, and we found that yes, zebrafish has both CREB-BP, which is also called CBP, and EP300, and you will see many other organisms here. This shows you the conservation across species also. Yeast also has a form of this enzyme, right? And humans and mouse and all that. So we then identified the genes that are there in zebrafish, and we decided to do the first thing that I talked about, knockdown. This slide now is familiar. We decided to reduce the levels of this protein by using antisense oligonucleotide. So we did that. I'm not going into detail. Just to recapitulate, these are the characteristic features of the patients. What do we see in fish when I knock this gene down? So okay, so it's a very complicated slide, so but I don't want you to look at everything. The first panel on the left hand side is a bright field image of fish embryos that have been injected with antisense oligonucleotide when they were one cell stage. So the antisense oligonucleotide would have interfered with making of EP300 or CREB-BP. And I am going to look at what happens to these fish when EP300 or CREB-BP does not work properly, right? So if can you see my mouse? Yes. Okay, so this is the normal fish. This is the eye here, and this is where the heart is, just below the arrow. And now compare the fish where the EP300 has been reduced. The head is smaller, the heart region is inflated like we saw in that compound treated fish. It's basically filling up with the water. It is much more pronounced in the CREB-BP antisense oligonucleotide injected fish, right? So there is a heart defect which is also seen here. Now I have used a transgenic animal with a red fluorescent protein expressed in the heart, and this is the ventricle that I am labeling here, which is highlighted here. So you can see that there is a difference in the size and the positioning at the heart chambers, which can account for the ballooning of the pericardium that you are seeing here. There's also this arrowhead here is showing you that there is a reduction in the jaw. So the jaw is a very specific or tissue that came up in vertebrates, and it is very important evolutionarily speaking for lots of things. Imagine not having a jaw. What kind of food can you ingest? Very soft food only, right? And very limited nutrition. Once you have a jaw, you are able to chew, and that gives you access to a lot more nutrition. That suddenly changes the trajectory of evolution. From other vertebrates to apes also, there is a dramatic change in the way the jaw is organized, and it is thought that that change so our jaw is not as powerful as some of the other organisms, right? So we need to cook food mostly to chew them and eat them. But why was that done? That was just so that you can make more space for the brain. So compromising on the jaw, you made more brain. So we can cook and we can hunt and we can do a lot of things, but we cannot eat raw food as efficiently as a dinosaur can, right? So, so vertebrates have jaw. It's a very important structure. I will tell you why I'm talking so much about it in the next slide. So these fish also have problem in jaw. So this green picture that I'm showing you is labeling all the cartilage in the head region with green fluorescent protein. The head region, craniofacial cartilage, is also the tissue that gives us our shape of the face. Every face in this room is different, right? Although we are 99.9999% same. Small differences in the control of genes on making the craniofacial cartilage makes our face so different. And you saw that the patients also have very characteristic facial features, right? So the jaw of the fish is also very dramatically affected when I interfere with the production of this gene. So the patients had craniofacial cartilage defects. The fish has cartilage defects in the face. The patients had heart defects. Fish also have heart defects. The patients had gut abnormalities, and so we decided to look at the neurons in the gut. So you might know that you've all studied that our gut has this peristaltic motion to move the food downward, right? How does that happen? That is because there is a set of neurons called the enteric neurons which control how the gut contracts, and these neurons are what we have labeled in red here, and they are completely absent when I knock down the gene again, right? So multiple tissues that are affected in the patient are also affected in the larva when I take this gene out, telling me that there is similar kind of genes are doing similar kind of functions in the development, right? So now I just want to sort of take a short turn to ask the question that we were interested in addressing. So we looked at all these features that are highlighted in red here in these patients and realized that all these tissues originate from a cell type called neural crest. These neural crest cells are a specific, very interesting kind of cell type that is only there in vertebrates, and it's very important for making craniofacial cartilage here, also for pigment, for enteric nervous system, for the peripheral neurons, for the sensory neurons. So lots of things that we consider very important for our existence comes from these crest cells. And these crest cells migrate long distances in the embryo to form these different tissue types, right? So we wondered if the crest is defective in these embryos when we removed EP300 or CREP-EP. So this is what the crest looks like in the zebrafish embryo. This is these cells are stained using an antisense oligonucleotide against a gene that is expressed only in the crest. Okay, it's very complicated sentence, but basically only the cells that form the crest are purple here, right? So that I can look at this without getting distracted with the rest of the embryos. Okay, so the question was, do the histone acetyltransferase genes play an important role in crest development? And for that, we used again a little bit of chemical biology. Instead of knocking down the genes, we took a chemical compound called C646. That is a compound that inhibits this enzyme function. right? Both EP300 and CREB-BP, both are inhibited by the same compound. So I can look at both together in one experiment. And don't look at all these embryos, just look at the last segment, which is 18 somite stage. You see that these streaks of cells on the back of the fish, these are very limited in the when I inhibit the CREB-BP EP300 enzymes, which means the enzymes are important for these cells to migrate this way, right? I can also do this experiment by knocking down using antisense oligo and doing live imaging in a fish which has fluorescently labeled neural crest cells. So that's what I'm seeing here. This insert here is showing me the streaks of cells that are migrating, which are absent when I knock down these genes. We also did CRISPR-Cas mutation of the EP300 gene. So using genome editing tools, we made a cut in the EP300 gene and inactivated this gene to see what it does. The fish does not survive. So we got some pictures of the embryo, but the fish line never survived because this gene is so important that the fish cannot survive long term without this gene. So, but we were able to see that, yeah, the same cells that we were looking at in other, in the oligo-injected fish was also absent or not migrating properly when you make a mutation. So basically, neural crest cell migration is affected. This was not known before from patient studies or cell studies. Nobody had thought about looking at it, right? In a patient, you cannot see whether neural crest is affected, right? The doctor is only looking at which all systems are down in the patient and how they can help the patient, right? So making a model in the fish, being able to do live imaging, labeling different cell types, perturbing them the way you want, we can ask questions to understand the development in such detail that we can identify such issues that there is a crest development effect. So we then asked, can I find chemical compounds that would reverse or prevent this defect that I'm seeing? So we did a small screen because we are looking at histone acetyltransferase enzyme and its mutation. We did a screen with compounds that inhibit such acetylation processes, right? Acetylation and deacetylation processes, and we found some compounds. So this is the control embryo that you're looking at here on the left hand side, and this is what happens when I knock down EP300 or CBP. You get crest severely limited. There is no streaks of crest that you can see, but you can actually see some streaks of crest in these fishes, right? These have been treated with a compound, so they have the gene knocked down. So the gene is not functional, but the chemical could rescue the problem that you saw. So like a drug, you are able to prevent the phenotype that or the defect from manifesting itself, right? So last couple of slides. So the question is, then I use zebrafish to create a model for human disease. I found there is a defect in this cell type called neural crest. So what? It's fish. How does it matter? Maybe it doesn't happen in human, right? I can't go and look at a human embryo right now. So as a surrogate, we got some cells from a Rubinstein thyroid patient and made a stem cell out of those cells, right? So we took some blood cells and we created an induced pluripotent stem cell from these patient cells, and then made neural crest of these cells in culture. Okay, so this is how the stem cells look from the patient. Then this is not important. So now these cells, when you make them into neural crest like cells, they sit like a ball in the plate, right? And then from the ball, cells start migrating outwards. Like the neural crest migrates in the embryo, in the plate the cells start migrating outwards. So if you can see this image here in T10, this you will see that there is a halo of sort of streaks of cells coming out from that ball in this well. That's basically that ball of cells and the migrating neural crest cells coming out of it. That's a normal on non-diseased patient stem cells. Below is the Rubinstein Tybee patient stem cells, where it is very difficult to see such streaking cells. There are some, but much less than what you see in the control. The next slide will make it even easier to see. So now I have fluorescently labeled the crest cells in green and red actually. So you can see that there are green cells that are migrating out, all these cells that you see alone here compared to the ball of cells, and this is the patient cells where there is very little migration. So I'll sum up the story that I showed you. Basically, we picked up these histone acetyltransferase genes that are mutated in a disease called Rubinstein Tybee and mutated them in fish. And we saw that what we see in fish are that the same tissues that are affected in the patient are also affected in the fish. Not in the same way, but similar tissues, same tissues. Then we showed that there are neural crest defects in these fish embryos that we did not know of before in this disease. And in the end, we were able to show that the human cells from patients also have similar kind of neural crest defects. So now we want to, of course, go forward and see whether the same chemicals can prevent that defect in the human cells or not. What is going on in these cells? Why are they not migrating? We want to understand all this. That's our next sort of. This is the lab over many years. And sorry, I took.
Thank you, Dr. Chetana. Please kindly use the mic for asking the questions.
I can stand here.
Very interesting and very informative. I have a couple of very simple questions. Like, when you induce this pluripotency in the last couple of slides, like how long it takes? Like, is it by injecting Yamanaka factors and things?
So
Can
not by
you give some-
injecting.
yeah.
So the way we so pluripotency means cells that can form many different types of cell types, right? But when I take blood cells from the patient, those cells can only form blood cells. They cannot form anything else. They are committed cells. So what I am doing is I am taking these committed cells, I am inducing stem cell like behavior in them by 4 factors that Dr. Radhakrishnan was saying, Yamanaka factors, because Yamanaka was the scientist who discovered them, got the Nobel Prize for it. 4 transcription factors that can be put inside these cells. So we put them by transfection. Those transcription factors go there and change the genome, like open up genome here and there, and give the cells the ability to become stem cell like. Now they can make all kinds of cell types. So I skipped that slide. This slide is showing that, so there are 3 genes showing here, GATA4, TUJ1 and encadrin, showing that these stem cells can now make mesoderm, endoderm and ectoderm, 3 different cell layers, while they came from blood cells which could not make anything else, right? So they have become stem cell like cells, and now I can make them go to a heart pathway, I can make neurons from them, I can make neural crests from them, I can do lots of things with these cells.
So,
Yeah.
sorry, taking a chemical route or when you
It was using transfection of the 4 transcription factors
Okay.
only, not chemical.
Okay.
No, genetic.
Yeah. So another quick comment is like, it is very interesting that you see the tailless fish, for example. So this is an observation which is almost 3 decades, like So when we had earlier, like it's also connected to the water pollution, and the fish is in fact very sensitive. Like, in fact, if you look at the bottles of the pesticides, you always see a fish and mark like it. So it is known to the chemical makers that it's going to affect fish varieties. It's rarely known. So one of the observation, maybe it's also benefit to the audience in large. So we see a lot of, so earlier there was a 3 decade back we had clear streams and you can see the fish and we can swim with them. So after a decade or so, these chemicals start coming, the pesticides. Then what we observe is that, what I observed 3 decades back, that many fish varieties have no fins. And they are live and they have no tails. So this is something which we grew up and we so observed, because the fish tank is the whole stream.
Right.
And then what happened is that it took a long time for me to figure out what actually caused it. Because I want to make the connection to the pathway which you have, which is conserved in many varieties. So most of the chemicals which is targeted the pests, it is targeted the muscle joining junctions of the wings. So the pests, the wings are targeted, and exactly what the analog of the fish is the fins.
Right.
So this is exactly going and binding to the same region which is conserved all species, and you start up seeing all the finless fishes in the thing. This is some observations which naturally with the plain eyes you can see over 1 decade. I just want to make the connection.
Thank you. That's a
thank you.
beautiful illustration of, if you keep your eyes open, you have experiments around you.
Yes, that's right.
And also that we think we are so different from the pests and flies, and what affects them will not affect us. But just like you said, again, the same genes do the same things in all of us, and you have to be very vigilant about killing an organism and thinking that you are safe from that compound. So thank you. Yeah. You had a question. Yeah.
Okay, my doubt was quite simple. In current situations or in current world, in what cases will you prefer forward genetics instead of reverse genetics?
Although I said with great arrogance that we know the human genome and we know all the genes, and now we should go backwards from the gene to the phenotype, the truth really is that we know so little of what happens inside us, right? So there is still a lot of space for starting from a phenotype to going to gene. So you might still, so every time I figure out what a disease, in a disease condition, which genes are mutated, I am going that way, right? It's nature's forward genetic screen. So we have mutants all over. We are all mutants, and some of us will have phenotypes, which can be neuropsychiatric, it can be diabetes, it can be cleft palate, anything like that. And each of us, actually you can work your way backwards to what the genes, what the mutation is, right? That is what genomic scientists do when you take patient samples and go back and do the sequencing. So that's a natural sort of consequence of where you would use the phenotype to genotype connection. But in a model organism also there is still, if I want to, for example, understand where do the GABAergic neurons come from? I know very little even now. So I can still use a forward genetic screen, make mutants and look for GABAergic neurons and say, okay, these fish don't have GABAergic neurons. So that means there is a gene that is important for these neurons, and I can figure it out. The identifying the gene has become very, very simple now, because I know the whole sequence of the genome. I can design ways of just looking at that gene and identify the mutation. I don't have to my generation scientists used to spend whole PhDs or 10-20 years figuring out what the gene is. It's basically a week or a month's job now. Yeah. So yeah, we are far from understanding everything. Yeah.
So I don't do developmental biology. What I look at
zebrafish is a ideal model organism for before going to rat models and before going to mice models. And I work in the area of cancer and Alzheimer's. So as I read a lot of reports where they are saying that zebrafish might be a upcoming model for Alzheimer's disease. How do you see it as a potential model organism for Alzheimer, looking at the complexities of the diseases in depression, basically psychological disorders? This is what I want to know.
Right. So there are two ways of looking at the model. You can model the behavior. You can also model the cellular effects. So in Alzheimer's and Parkinson's, we know neuronal death is important. We could use the fish as a model to study the neuronal death, which is beautifully done because of the transparent nature of the embryo, and you have markers to light up specific neurons, and you can also see cell death in live fish, etc. You can also do behavioral studies. You said depression and Alzheimer's. So, for example, I showed you that there are assays for anxiety and also depression. So these are correlates. Okay, I would not say that the fish is going through. So if I was mimicking, say, schizophrenia in a fish, I would not look for the same phenotype, of course. I would look for similar kind of, or its sort of surrogates in fish. What does it mean for the fish to have these particular neurons die? What kind of effects are there on the behavior? I would make that correlation rather than go and see whether they are having issues with perceiving reality or hearing voices. That's not what I would assay there. I would assay that is there a way in which do they swim differently? Do they socialize differently? I would look at things that are more relevant to the fish. But so you have to when you create a model for another organism in fish, mouse, wherever it is, you should not be looking at the same thing that you see in the patient or the human. You should look for correlates of that, is what I feel. I mean, it's very important. Like even in something like craniofacial cartilage defect, the patients of Rubinstein IB have a particular kind of nose or a particular kind of thumb. You are not going to be able to see that in fish, but you can see pectoral fin defects. Actually, I did not point that out. The equivalent of our arm is the pectoral fin in fish, and there is defect in that. So you have to make that correlation and use that as your assay rather than trying to mimic the human phenotype. Yeah.
So my next question is, are you open for collaboration?
We can talk about it. Yes, of course. My interest is neurodevelopment, so in that purview, yes.
Thank you.
Is what makes the antisense oligonucleotides resistant to enzymatic degradation?
Okay, thank you for asking that. So the oligonucleotides that I showed you, they are not plain simple RNA or DNA molecules. They are actually modified oligonucleotides. So you must have seen MO written in all my slides. These are morpholino modified oligonucleotides. So sugar group in that backbone is different from the sugar group that is there in our natural RNA. So the enzymes that degrade RNA in our cells, the RNases, are not able to degrade these, which doesn't mean that they will stay there forever. So the observation suggests that 3-4 days is the lifetime of these oligonucleotides that we inject, but they stay for that long. They are more stable than the natural RNA. If I injected a natural RNA oligonucleotide, it may get degraded much faster than these modified.
I have another question. So the chemical screen zebrafish includes a the identification of small molecules like which is an IGIB inhibitor. What are the key challenges in translating this derived chemical genetics into the mammalian?
Okay, so yeah, so we have tested that molecule in mouse models of fibrodysplasia ossificans progressiva. That is a disease, FOP, which if you have watched this movie called Samuel Jackson's movie, I forget what it is called. It's in which basically the bones become stiff. So the muscle and other tissues in your body slowly ossify. Okay, so what happens is we have muscles here on the back, and then there are bones, vertebrae, right? But suppose slowly that muscle all become ossified, because becomes calcified, and the muscle is not able to contract anymore. What will happen? We will have a stiff body, right? And you can imagine if my diaphragm muscles stop functioning, then I cannot breathe. So FOP is a very painful disease in which slowly, progressively, the patient loses mobility and then dies because of suffocation, because of the failure of diaphragm muscles. So this disease is caused because there is overactivation of BMP signaling. So in this disease, so we created a model for this disease in mouse, and we used dorsomorphin injections in them to show that there is actually reduction of ossification. You can inhibit the ossification in them. There is so you have to have a mammalian model first. You have to take it to that stage before you go anywhere near human. Then there is of course toxicity and all that. But so one of the challenge I would imagine is that in fish, delivery of a molecule is very easy. Just add it to the water. You don't have to do anything else. That's not how it's going to work for a patient. So you have to convert a water soluble or a skin absorbing molecule to something that can go through your gut or through injection. So the mode of delivery would be different. So which will also change your the stability of the molecule in your body. Whether whether human beings have enzymes that would degrade that molecule faster. So for these, any drug molecule has to go through a screening called ADMET. Basically, absorption, degradation, metabolism, I forget the toxicity. So these have to be tested for any drug molecule. So similar kind of things would have to be done there also. So something you find in zebrafish, you cannot directly jump into human and imagine that it will work there with the same efficiency. There will be many steps, and it will fail also in many steps, which is part of drug development. That's a natural part of drug development process.
I have one last question.
Yeah.
So earlier you mentioned that if we cut all the blood supply to the growing tumor, it may die. But which is the blood is basically responsible for supplying the growth factors for the tumor to grow. So, but it has been studies have found that even if the IGIB growth receptors have been cut off from the tumor, the tumor still persists due to autocrine signaling. So how do we overcome that challenge?
Yeah, so I am sure there are cancer biologists in the audience who are more equipped to answer that question. So yeah, cancer is very smart. So you figure out one way to kill it, it figures out another way to sort of continue growing. So, but that challenge is the same for all cancer drugs. And I'm not going to go into that area. It's not my expertise, and I will say some stupid things. So yeah.
Thank you.
I wanted to ask a very basic question that being in educational institutes, when we think of having any model organism, we think to work with any model organism, we're worried about UGC norms which are prohibiting dissections. So can you guide us tackling this legislative things?
Right, so the UGC guidelines against dissection, if I understand correctly, with very little knowledge myself, is that for studying, for demonstrations, you should not use invasive techniques. So for research you can. And so the whole spirit of prohibiting an animal's dissection in the school was that we do not take animal experiments lightly. It is not something, I am from a generation where we did animal experiment dissection in school, and it was very callously done. We did not appreciate that you're working with an organism, right? So for any kind of educational or research institute, now there are strict ethical guidelines which have to be followed. You have to first get approvals for a project before you even execute an experiment, and you have to follow those protocols strictly so that you are treating the animals humanely, you are not wasting them, you are not unnecessarily doing an experiment. So these are all in the spirit of the fact that animals are not something that you just pick up and start fiddling with. These are creatures and you give the respect to those. So if you follow those guidelines, which there are, it's all well sort of written down. Even in educational institutes, we can use the use organisms for experimentation. Then there are different kinds of experiment, right? Behavioral experiments, observational experiment, and interventional experiment. So again, you can decide whether intervention is necessary for what you're doing or not, or mere observation is enough. Even observation itself is a big jump from studying something from textbooks. So having said that, I mean, as long as we follow the guidelines and we get the necessary permissions, it is possible in educational institutes also.
If there are more, I think we'll have to stop now, and we will definitely appreciate more questions later. There is going to be a student interaction with her at 2:30. So you can keep all your questions for that session, and it's a good 1 hour session for you with her. So please do make yourself available and ask her all the questions you have, but we'll have to stop the session now and continue for the next part.
I now request our Pro Vice Chancellor, Dr. Mode, to felicitate Dr. Chetana Sachidanandan. I now request Dr. Chiza Jacob to give the vote of thanks.
Thank you everyone. This is wonderful to see that we have so many students in the audience and a very interactive session. I would like to thank, firstly by thanking our Chancellor and the Vice Chancellor for inviting Chetana and giving us an opportunity to listen to her work and introduce the model organism Zebrafish to our audience. On behalf of the Somaiya Vidyavihar University, Chetana, we thank you for accepting to come and deliver the Somaiya public lecture. And thank you so much for spending a lot of time and explaining right from the very basics to the use of chemical mutagens, the chemical screens done, use of the latest genetic tool, that is CRISPR-Cas9, and how you can use fluorescence microscopy to answer and look at different aspects of development and also look at behavior in zebrafish. This was really very informative. I think the students have definitely going to gain a lot of it. And also, very nicely, you ended the whole session by giving an example where you use all these tools in zebrafish. Thank you once again. I would personally like to thank all those who helped for the smooth conduct of this session. The staff from Aurobindo office for the support in booking the hall, Yugalji, Maheshji, Shettiji and Pratik. From the VC Sir's office, Aparna ma'am, Jensa from the PR office and the IT team. A big thank you to Hemant for helping with organizing the travel, the food, the sound system, the photography, and all details needed for the smooth conduct. I would also like to thank Divya, our BSMS student, and Christina. Divya agreed to be the master of the ceremony and stood here next to Chetana as long as Chetana stood here. Thank you so much. And thank you all for coming and participating in the session. Thank you also for people who have come from outside Somaiya University. Thank you, ma'am. The former principal of Guru Nanak College, Sion, for coming and being a part of this session here. And once again, a small reminder to all the students, the UG, PG and the PhD students, you will have an interaction session with Chetana at 2:30, and it is a 1-hour session, so please be available and you can come and interact with her and talk to her. I would like to say thank you to all of you for being here. Thank you once again, and see you all the next time for the public talk. Thank you.